Hi again. So today's lesson focuses on not real examples, but examples of transcription and translation working with sample sequences of DNA. So I'm just going to basically walk all of you through one example, and then I have a worksheet for you to do today in class. When you finish your worksheet, upload your answers to Canvas, and that's it for today. So let's talk about the whole idea here. On the screen, what I've given you is a sample of DNA. And you can see the 3' end up top, 5' end down below, and then to the left I've marked here promoter region, which is supposed to tell you that if the promoter is located here, that means transcription must start over there on the left, and then it will progress from the left to the right. So first of all, without me telling you, see if you can identify, and pause the video if you have to. Sorry, there's a motorcycle. See if you can identify which one of these two strands is the template. Pause. I'm pausing. Okay. So the template is going to be on top here because the template strand is read from 3' toward 5', because all polymerase enzymes build new strands in the opposite direction, from 5 to 3. So when we go and we make our mRNA molecule, it's going to start with a 5' end because it's reading the 3' and making a complementary 5' strand. So the first base is going to be C, the next base is going to be A, the base after that is going to be U. Remember, there's no thymine, we're only going to have uracil, and then G, C, A, blah, blah, blah. You get the idea. So there's phase one. You're going to find out which of your strands is your template strand. Then you're going to make a complementary mRNA strand. So there we go. You finish this all the way to the end of what we're looking at here. So this is a little snapshot of your worksheet, and this is the same sequence that I was just giving you. It only has the template strand there, so that makes things a lot easier. This is the full mRNA molecule, which would be produced during transcription. So that's right there. And then what they've done is they have broken down the codons, which would be produced from that mRNA strand. And I want you to take a second and take a look at the first 3 letters, the very first codon. Okay? Now notice that there's a single base missing. And if you know why that is, congratulations. If not, let me just explain. Remember, every gene has to start with a start codon, and that start codon is always going to be AUG, which codes for methionine, which I'll show you in just a second. So the first thing you need to do when you're approaching this problem is to identify where that start codon is located, and here it is, AUG, and that means that that first letter, that C, basically gets cut off. It's what we call non-coding information because it does not actually give us instructions for polypeptide synthesis, right? So that C is gone. We start with AUG. So going on from there, we basically break the sequence down into 3-letter words called codons. So we have AUG, CGC, AUA, and then we've got UGG, CUG, and then UAA. And now notice, again, there's this extra G on the end. And since that one letter by itself, that one G, does not make a full codon, 3 letters, then it also does not get translated because that's not enough to make a full codon. So that G gets cut off, and so we end here with UAA. The next step in this problem is they're going to ask you for the anticodons. And remember, the anticodons are what are on the tRNA molecules, which must be complementary to the mRNA codon. So AUG is complementary to UAC. And then you could just go from there. And that's that. So the anticodons are basically just complementary to your codons. That part, I think, is pretty easy. So you should have no problem there. The next step is where things get just slightly more complicated. And so I need to bring up basically another worksheet when I'm doing that. So what we're going to do now is this is the actual translation process. This is, transcription is done. And now we're going to translate that mRNA transcript into a polypeptide. So we need to figure out what these codons actually code for, which amino acids are going to be made in the protein here. So we have to look at basically a table that tells us exactly which amino acid AUG codes for. And the table looks like this. Let me pull one over into the frame so you can see it. All right, so remember our very first codon was AUG. So what you're going to do is you'll reference this table, and this will all be in Canvas. And you look at the first space is A, the second is U, and then the third is G, and you basically find it in the table. Here's AUG right there, and that codes for the amino acid called methionine, which is abbreviated here, it's just MET, and you can of course use the abbreviations. Now, just as a word of warning, you will never ever have to memorize this table. That would be a waste of your time and my time, so don't do that. You just need to know how to use it. So the first codon was AUG, which codes for methionine. So you would basically write MET, or they wrote the full name out here. You don't need to do that. You can abbreviate MET for methionine. Now our second codon, don't go to the anticodons. The second codon is CGC. So go back to that table. Here's the table. Oh god, I've already forgotten what was it. Where'd it go? CGC. So now we have to find CGC. We've got a C. Over here is G for the second letter, and then C. So CGC is going to give us arginine, which is ARG. And back in that worksheet, they've written out arginine. So we can just go with ARG. So we have MET and then ARG, and then you just keep going from there. And so every codon will correspond to one of those amino acids in the table. You write the full sequence out, and that's it. So it's just basically to get you to practice starting with some DNA, transcribing that into mRNA, breaking it down into codons, making sure that you leave off any extra bases on the front or extra bases on the end. So you start with the start codon, and then you basically just go through and you translate that into an amino acid sequence. Now there's one thing I need to warn you about before you get going on your own, and that is the concept of a stop codon. Now I mentioned this in lecture, but here's how it actually relates to your proteins, to your genes. There are a few different sequences, or a few different codons, which will give you a stop. Now, when it says stop, that is not an amino acid. It's basically just the end. So if you get to the sequence UAA, for example, you're not going to add in an amino acid that we call stop. It's just done. So whatever the amino acid that came before it is, that's it. That was the last one, and now you've stopped. So if you do get to a codon, you write down in your sequence, it's a codon that says UAA or UAG or UGA. Those are the three different ways that you can stop protein synthesis. And so for that particular amino acid, you would just, there'd be nothing there. So in some cases on this worksheet, you'll only have like maybe 2 or 3 amino acids present, because if the fourth one is a stop, it won't be there. So you'll only have 3 for that example. I think that's it. I think the rest of this you can probably wade through on your own. And so, like I said earlier, you're basically just going to work through all of the questions on this example. Fill out the mRNA, the codons, the anticodons, amino acids. Page 2 of the worksheet is the same thing, except right here they don't, give you all the titles. They just tell you mRNA, and then tRNA, and then polypeptide chains with the amino acids. So it's the same thing all the way through to the end. And then when you finish, you can submit your work onto Canvas, and that's that. I think it should be pretty easy for you. And this is the last thing we'll do before break. So if I don't see you later in the week, have a nice break, and we'll come back and finish up all of our nucleic acid stuff in January when we get back. So that's it. Good luck. See you next time.